Welcome everyone. Let us look at what we call the microscope. We have already looked at what we call the hand lens, and we saw that basically the hand lens is used to make things bigger, and that is what we call magnification. Okay, the hand lens enables you to see things in details that you wouldn't otherwise you wouldn't have seen with your naked eye. When we say naked eye, we basically mean an eye that is not aided. So if we say that the eye is aided, it means that that eye is having a device that is enhancing it to be able to see things in details that it would otherwise not have been able to recognize. So that was the hand lens, and we talked about the limitations that it has. Therefore, that means that we should be having another, so that therefore means that we should be having another device or another equipment that we can use to see small things that we couldn't be able to see with the naked eye or even with the hand lens. So that means that we are now moving to more advanced machines that can be able to help us to see things in detail. And in here, we are basically looking at the microscope. And in this session, we are going to see the types of microscopes that we have. And then we'll look at, specifically we shall focus on the light microscope. And we shall look at the different parts of the light microscope, their uses, and how we can actually use the light microscope. And then finally, how we can make sure that we keep the light microscope in good working condition. That is, how we can care for the light microscope. So that's the discussion that we're going to have. Let's start by looking at a scenario where water in a pond looks cloudy and smells unusual. Your teacher assigns you the task of using a light microscope to investigate a sample of the pond water and determine what might be causing the issue. You collect a small sample in a vial and bring it to the lab and set up the microscope to identify any microorganisms or particles in the water and report your findings. So describe the correct steps of preparing a wet mount slide for the pond water sample and explain why each step is important. Why is it recommended to begin with the lowest magnification when observing a new slide? Explain the difference between coarse focus and fine focus knobs and when it should be used during the investigation. What features of the light microscope help you see the details of the organisms? And then the eyepiece of your microscope has a magnification of 10. If you switch to the objective lens with magnification 40, what is the total magnification of the image you're viewing? Show your working. So let's proceed to see how we can address these tasks. In this is used to magnify, very small specimen such that we can look at this specimen in details. So remember, magnification is when we have a small specimen, but when we are using a microscope, we can be able to see it as big and see the details of that specimen. So that means that we can use the microscope to see things that we can't see with our naked eye. So we can see cells, we can see tissues, we can also look at organs in details. Okay, and we have two types of microscopes. The first one is the light microscope. The second is the electron microscope. Okay, now we're going to see the difference between the two. So starting with the electron microscope, just like the name suggests, it uses what we call electrons. It uses a beam of electrons to magnify specimens. So that means that electrons have to pass through the specimen, and then we get an image of the specimen. And this image is a magnified one. So the electron microscope can actually give you greater magnification, about 1 million magnification. Remember, when we have X, okay, when we have a figure and then we have X to it, in biology, it's about the magnification. That means how big the image is compared to the specimen. Okay, so that means when we say 1 million X, it means that the image is 1 million times larger than the specimen. So that means that the light microscope is actually very, very, very good in seeing very small things that we want to study. Otherwise, the challenge with it is that it is quite expensive. However, very big research centers can actually use them. Okay, so that means that we may not find electron microscopes in your schools, in your school lab, but if you go to well-funded research centers, you will find electron microscopes there. So what you are most likely going to find in your school, or even at the nearby health facility, health center, or even hospital, you are likely to find the light microscope. Okay, so this light microscope, for it, its magnification is not as great as that for the electron microscope. So the magnification can be to 1,000. Okay, 1,000. Remember, the previous one was at 1 million. Then this one is at 1,000. The other difference is that you actually use light to see the object. Okay, that is basically why it is called the light microscope. So it uses light to illuminate and magnify the tiny objects. That means that if you don't have light in a light microscope, you'll not be able to see. What you see here is the diagram of the light microscope, and we are going to look at the different parts and what they are used for. So we have what you call the eyepiece. Sometimes you can have it named as the eyepiece or the eyepiece lens. So the eyepiece lens is what you see here. That's the eyepiece lens. It's actually where you place your eye. It plays an important role in magnification. So with the light microscope, you have another lens, which we call the objective lenses. We are saying objective lenses because if you see here, there can be 2 or even 3 lenses there. So that's why we call that objective lenses. Okay, especially for a compound microscope. So that means that you have 2 lenses that can play an important role in the magnification: the eyepiece lens and the objective lens. Okay, and then this part here, there is the body tube. All of that is the body tube, okay, which is that, and then the rotating nose piece, where the objective lenses are placed. That is the rotating nose piece. And then we have knobs. They are of two types. Knobs are if you hear of the word knob, it means this is a piece that you can actually adjust. Okay, so we have two knobs: fine adjustment knob and then the coarse adjustment knob. The coarse is the one that appears bigger on the microscope, so it is this one here. And then the one which is small is the one we call fine. So in biology, or generally in science, when we say fine, this one here, it means it is something that is small. That's what it means. And then we have the stage, which is where we place the slide, that has the specimen. So the specimen is placed on a slide. Let's look at another image here. Okay, so on the stage, which you can also call the mount, by the way, is where you place the slide. Okay, and we shall see what the slide is. Okay, so you have the mirror, which basically is down here. It reflects the light onto the specimen such that you can visualize the specimen better. And then, so you have the stage. On the stage, you have the stage clip. And then the stage clip is the one that holds the slide that I talked about. It holds it in place. And then the condenser is to make sure that the light is concentrated onto the specimen. That means that when you have the light being reflected onto the specimen by the mirror, the condenser is the one that concentrates that light onto the specimen. And then the diaphragm controls the amount of light that enters. So that means that it is an aperture. It's a hole that allows light to go through. But so it allows either little light to go through or more light to go through. And then we have the hinge screw, which is here, which allows you to adjust the level of the stage, such that if you move the level of the stage, it means you're moving the specimen, such that it is in a good position for you to be able to focus. And then this is the base, which you can also call the stand. It's the one that provides stability. Okay, the microscope. That means that you can put it on table and it doesn't triple over. And then the arm, which you can also call the limb, so is what you use to hold the microscope if you're going to move it. So you hold the arm and then the